Yeah, today it's my great pleasure to introduce Sam Boshar. Sam is a machine learning research engineer at Instadeep, where he works on large-scale genomic foundation models for sequence-to-function prediction and design of regulatory sequences. He completed his master's in computer science at MIT in Manolius Kelly's group, focusing on genomic language models. So we're very excited to hear about Nucleotide Transformer version 3 today. So with that, Sam, please take it away.
Thank you, Johannes. Yeah, and thank you guys for having me. Very excited to share our latest work, Nucleotide Transformer 3. Um, and yeah, I guess with that, I'll get going. I'm totally fine if people interrupt and ask questions, but otherwise, I guess we'll have some questions at the end. Um, so let's see. Yeah, so I guess as a bit of an overview, when we were brainstorming the directions of NTV3, we kind of saw the state of the field as the following, um, as being kind of separated into these three verticals. So we have sequence to function models that take sequence and predict some attributes of the sequence. We have these representation models, which are pre-trained on large amounts of data and can be easily fine-tuned to perform well on a variety of tasks and generative models. Um, and although there are models that kind of bridge two of these three pillars, um, there aren't really any that bridge all three of them. And we looked at all like these three pillars as being complementary of each other. If you have a model that learns good representations, that model should be better at sequence to function. And maybe if you can leverage the functional data, you can inform your representations and you can also build a better generative model. And so that was our idea with NTV3. How can we kind of unify these three approaches and get the best of all worlds? And so, yeah, I just want to talk a little bit about our goals in each of these three pillars. So at the beginning, we kind of set out some ambitious goals. On the sequence to function side of things, we wanted to be able to process 1 million base pair sequences at base pair resolution, because we believe this to be important for kind of long range interactions and also to have the base pair resolution for variant effect prediction and for, like splice site detection, et cetera. We also wanted the model to be multi-species, so to go beyond just human and to start generalizing to other species and plants, other animals and plants. And we wanted it to be kind of state of the art. And the final point was we wanted it to be easy and efficient to use so that people could actually use these models. And we've put in a lot of work to make our models public and very easy to use and also very efficient. So you can run most of them on just your laptop. The second point is on representation learning. So we wanted to go beyond previous methods, and in addition to doing a large scale pre-training on like genomic sequences, so we trained on over 10 trillion tokens or 10 trillion base pairs and 125,000 distinct genomes. We also wanted to fuse functional and annotation information in this stage as well. So not just to predict it, but also to use it to inform the representations. And finally, we also wanted to make the representation species aware. Um, and we'll talk more about this later. Then finally, on the sequence generation side of things, we thought there were kind of a lot of low-hanging fruits where we could improve on this side, like on this pillar in particular. So first, we wanted native in-context generation. So we think it's important, especially or a lot of DNA generation tasks, you want to be informed by the context on both sides of the sequence, sort of this in-painting. And this is something that like autoregressive models struggle with natively, at least. We also wanted sequence generation to be steerable with flexible conditions. So we want to be able to generate conditions or generate sequences conditioned on this annotation and functional information that we had trained on. The third point is an important one and kind of goes back to what I was talking about in the first slide, is we want to be able to leverage the pre-trained and these post-trained models, which I'll discuss more. So we didn't want to have to retrain this sequence generation model from scratch. How can we leverage the rich representations we learn in pre-training? And then finally, we wanted efficient generation. So to be able to generate sequences efficiently up to 1 million tokens. So yeah, excited to introduce our latest work, and we managed to hit on most of these, if not all of them. And so first, I will give just a quick outline of where I'm planning to go. So I'll talk a little bit about the pre-training first, then the post-training, which is the stage where we introduce functional data, and then I'll dive into the fine-tuning and the generation and have some closing remarks. And again, feel free to stop me. Otherwise, we'll have some questions at the end. So first on the architecture. Like I mentioned earlier, one of our main concerns in the design stage was to build a model that was easy for the community to use. So we settled on a unit architecture, which is, I guess, not unique to NTv3. And we have basically a standard unit with 7 convolutional layers and skip connections. And this allows us to process 1 megabase pair sequences of DNA uh, at single nucleotide resolution, and to be able to do this on basically your laptop, which is pretty cool. Yeah, so just to compare to some other foundation models, um, basically nucleotide transformer is a lot more efficient than the competition. Obviously, if you have kind of this full transformer, it doesn't extend to these long sequence lengths. But we show that even the largest NTv3 model is much more efficient than, say, Caduceus, which is, 60 times smaller. So now I wanted to give a quick overview of uh, the training stages of NTv3, so the different phases. So we began with pre-training on the Open Genome 2 dataset on over 9 trillion tokens from 128,000 species. And this was just simple mass language modeling. We then introduced the second stage, which we call post-training. So in post-training, we continue to do uh, this self-supervised training. We continue to have the mass language model objective. But in addition to this, we also add um, like functional data and annotations on the sequence, and we compute a loss on these as well. So we'll have various assays as well as annotations. Then we take this post-trained model and we evaluate it in 2 regimes. So the first is fine-tuning. In fine-tuning, we want to determine a few things. First, how good is the post-trained model? How easily can it be fine-tuned on a variety of tasks? And 2, does it help to post-train? Does it outperform the pre-trained model? And then the second direction we look is, how can we take this post-trained model and apply a very lightweight fine-tuning to turn it into a generative model? And does it help to have done this pre and post-training first? And so these are some questions we'll answer. So quickly, just on the pre-training. So as I mentioned, we did pre-training on the Open Genome 2 dataset. And one thing that Nucleotide Transformer employs is this mixed sequence length curriculum. So we pre-train in 2 phases. The first phase consists of the majority of the tokens. It's almost 11 trillion tokens. And in this phase, we pre-train on short sequence lengths for efficiency. So the sequences from 1 to 8 kb that are mixed into the data set. So the model sees 1, 2, 4, and 8 kb sequences. After this first stage of pre-training, then we do a sequence length extension phase where we extend the context up to 1 megabase pair. And we do this in 7 consecutive phases, but basically at each stage, we introduce a longer sequence length and continue to mix in all of the shorter sequence lengths. And we found in our own ablations for this to be important, for it to be important to see all sequence lengths. And that these unit architectures are very susceptible to forgetting how to process the short sequence lengths um, if you don't continuously mix them in. So this is one thing that we introduced in pre-training. So on the left, gives an overview of the data distribution. We basically pre-trained on the raw data distribution of OpenGenome 2 with some exceptions. The main being that we waited to introduce the whole eukaryotic genomes until later stages of pre-training. So the initial 8 kb and 16 kb stages of pre-training uh, don't include the whole genomes. And we reason that since like this allows the model basically to focus on some of these more functionally enriched regions. And then once the sequence length gets long enough, then we start passing in the whole genome sequences as well. And on the right is just a particular subset of the OpenGenome 2 dataset. And you can see like over the course of pre-training, performance like improves across these sequence lengths. And generally, we achieve the best performance at the longest sequence lengths, which shows that like adding the extra context is useful. So now to talk a little bit about post-training. So after we finish pre-training, we take the final length extended model, and this model now undergoes post-training, where we're simultaneously training on a sequence, an annotation, and a functional loss. So the sequence loss continues to be this mass language model objective, BERT style. On the annotations, we have a focal loss, or basically a cross entropy. And on the tracks, the assays, we have this Poisson loss. And basically, the reason we introduce this stage is we reason that that simultaneously training on these annotations and these tracks can inform better representations of the DNA sequence as well. And we evaluate this later on the various like fine-tuning benchmarks. So yeah, a little bit more on the post-training. Um, on the left, you can see, or I guess in the middle, you can see a breakdown of the different species. So we trained on 24 species. Among those are human, mouse, and a variety of plants. And each of these species had like a large variance in the number of functional assays. So human alone accounted for almost half of the functional assays, followed by mouse, and then a variety of plants. Um, so in post-training, we didn't sample each of the species uh, equally. We sampled human more often because there were more tracks, and then mouse, or then the remaining of the animals. Um, and you can see on the left the breakdown. So some species only have annotations, some have functional tracks plus annotations, and um, and then we distinguish human from um, the remainder of the animals with functional tracks and annotations. Because first, I guess because we were concerned with the model performing well on human, and also because there's a lot more functional data for human relative to any other species. So yeah, I guess now I'll share just some qualitative analysis of the predictions of this post-trained model. So here we're looking at a single cell attack-seq from Drosophila, muscle cell development, and we can see that the NTv3 predictions accurately recover experiment. And here's another example on leukemia. We also can look at the functional annotation head and see how well that recovers experiment or recovers annotation. And here we can see that NTv3 accurately recovers the genes, introns, exons, splice donors, splice acceptors, etc., and can do so at 1 million base or at 1 million base pairs. This is just a zoomed up version so that you can see the structure of the gene. In post-training, we also found it useful to continue this mixed sequence length policy. Um, so on the right side, so this slide shows comparison between Borzoi and nucleotide transformer. On the right side, you can see how both models perform across a variety of sequence lengths. Um, and you can see that by training with a mixed sequence length policy, NT mitigates this degradation that happens at short sequence lengths. So Borzoi didn't have this policy, and they suffer a lot at the shorter sequence lengths. Um, on the left side, you can see the results at, I think, 512 or 524 kb, which is um, the max length, which is the length that Borzoi processes. And we can see that it matches or exceeds in all of the major assay groups. We also evaluate on genome annotation as well. So you can take the post-train model and use the annotation head and compare it to existing methods. So if you compare it to our previous work, segment-NT, we outperform across all species and basically across every genomic element. So this is pretty exciting. We also see that when you extend from 30 kb to 1 megabase pair of context, that tends to help as well. So the model's using the extra context and is outperforming a previous state-of-the-art specialized model on this task of genome annotation. We found that in general, it helps a lot that pre-training and post-training improve the performance of genome annotation a lot. Finally, we can take this post-trained model and we can interpret it. And the nice thing about NTv3 is since it processes so many modalities, we can get a sense of like a more holistic picture. We can ask questions like, do the different modalities agree with one another? So here we're looking at the HEB1 gene, and we can see that the annotation prediction head aligns with the tracks. And we're seeing like accessible regions where the enhancers are, and the expression also aligns. And now we can do some kind of in silico interpretability experiments. So on the bottom, like right, you can see an experiment where we knocked out all of the enhancers and introduced these enhancers one at a time. And we look at the log fold change. And this we found that the log fold change of introducing these enhancers one at a time, the magnitude of this recapitulated the experimental values. And so this gives us some insight that NT is able to not only capture these enhancers, but also capture the importance of each of these enhancers. So now that we've discussed the post-training, we can talk about fine-tuning. So in fine-tuning, we want to take these post-trained and pre-trained models and evaluate them on a variety of downstream tasks. And in particular, we want to ask the question of whether this post-training was useful. Does it help at all? Does it yield improvements over pre-training alone? And so I guess to accomplish this, we introduced what we call the NTv3 benchmark, which consists of 106 multi-species longish range tasks. So these include gene expression, DNA accessibility, genome annotation, and we evaluate everything at single nucleotide resolution. We also make a point to have evaluations that contain species that were not seen in post-training, just to control for this as well. So we've condensed these 106 tasks into by species, and you can see that the post-train model tends to perform that overall NT models outperform the competition, and that the post-train model consistently outperforms the pre-trained model. And this is consistent for held out species like tomato and chicken, which were not seen in post-training. So this gives us some confidence that this post-training stage is not only improving the representations, but it's allowing better generalization for species that were not even seen in post-training. So we're just looking at the post-trained model compared to the pre-trained model of the same size. On the left side is kind of the win rate of the post-trained model over the pre-trained model. And you can see that across basically all downstream tasks, the post-trained model outperforms the pre-trained model, which is pretty cool. Um, on the right side, we also look at efficiency of these models, training efficiency. So in general, the NT models are very efficient to train, both in training time and number of tokens needed. The post-train model in particular is very efficient and requires almost no like fine-tuning tokens um, relative to the like even the pre-trained model. We also evaluate on some aggro tasks, and we find that all versions of NTv3 outperform our previous aggro model, which is much larger, aggro-NT. So aggro-NT is a billion parameters, and here, even surprisingly, it's being outperformed by the 8 million version of NTv3. We also see another, like the same pattern here, which is that the post-trained model continues to outperform the pre-trained models, and that increasing in model scale yields better performance. Also, that generally, that increasing the model context leads to better performance as well. So now I'd like to talk a little bit about the generation experiments. So ooh, I guess just a bit of an overview first. Um, when we looked at what existed, well, what methods existed currently, we had goals. We saw a few areas where we thought things could be improved. The first, as I mentioned, was this native in-context generation. So a common design task is to be able to infill a region, say some functional region, given the context around it. And we wanted a way to do this naturally. The second is we wanted generation to be efficient and scalable. If the model can produce or can process up to a 1 megabase pair, we wanted to be able to generate sequences up to a megabase pair efficiently. If you kind of naively use autoregressive approaches, this might yield something like a million model evaluations, which is obviously not very scalable. Um, the third point is we wanted a method that naturally extended the pre and post training. So we wanted to be able to leverage the pre-training and the post-training and the representations we learned and do just maybe a lightweight fine tuning to turn the model into a generative model. And then the last point is we wanted the ability for precise control and flexible conditionings. Um, and we wanted to be able to condition on kind of a variety of aspects about the sequence and generate sequences that adhere to this conditioning accurately. And classifier-free guidance allows us that precise control. So we went with a framework called Mass Diffusion Language Modeling, or MDLM, because it accomplished these kind of four goals we had set out. If you look at the in pre-training, we basically do this BERT noising and a reconstruction with a cross-entropy loss. The reason MDLM is so nice is because it naturally extends this framework. So MDLM is basically just a weighted cross-entropy over, over a variety of noise levels. So in this framework, you first sample a noise level, noise the sequence to that level, and then you weight a cross-entropy loss on the reconstruction weighted by the noise you sampled. And so since the objectives are so similar, you can take the pre-trained base and do a very lightweight fine-tuning with this mass diffusion language modeling. And you get a model that is generative and has kind of principled foundations. So this is exactly what we did. Um, and to evaluate NTv3's generative capabilities, we really wanted to validate this in cellulo. So we proposed two experiments, and Both are conditional enhancer generation experiments. So we're looking to generate enhancers. The first is activity condition enhancers. So here we're basically asking the question, can we generate an enhancer with a specific activity level for a fixed promoter? So in the context of one promoter. The second challenge is a bit harder. Here we're asking, can we generate promoters or can we generate enhancers that are specific to a promoter? So we have promoter A and promoter B, and we want to generate enhancers that are selectively active in the presence of one promoter, but inactive in the presence of a background promoter. So these are the two experiments we propose. I guess this is just an animation, I guess. So you can see NTv3 being prompted by the activity level, and here it's infilling some regulatory region. So here are the results. So for the first experiment, we find that NT enables state-of-the-art activity-controlled enhancer design. So we prompt with the activity level we desire, and we generate the enhancer, and we find that it aligns very well with the native enhancers of each level. And we're even able to generate enhancers that exceed the sort of highest or the highest level of the natural enhancers and enhancers that are even less active than the lowest level. And this was validated in Cellulo. For the second experiment, we wanted to verify whether NT could generate promoter-specific enhancers. On the left side, you can see our in silico results. So these are kind of the predictions that we made, or these are like this is the forecast we had for how well the sequences would perform in Cellulo. And the basic design of the experiment is we had two promoters, this RPS12 and DSCP. And we prompt NT to generate sequences that are specific to one of these promoters. And we do this by asking it to generate sequences that are maximally active in the presence of one promoter and minimally active in the presence of the other. And you can see that in our in silico results, when we ask NT to generate sequences that are RPS12 specific, which are the light blue, these tend to have much higher activity in the context of RPS12 than they do in the context of DSCP. And vice versa, when we ask NT to generate sequences that are high activity in the context of DSCP, generally have higher activity in the presence of DSCP and lower activity in the presence of RPS12. On the right side, you can see the in cellulo or in vitro, I guess, sorry, in vitro validation. So the first bar plot shows that when we ask sequences to be like high activity in DSCP, they're on average twice as active in the presence of DSCP than they are in the presence of RPS12. And on the furthest right, we do the reverse experiment. And we find that when we ask the model to generate sequences that are high activity for RPS12, they're 4 times more active. As a control, we also used an Oracle-guided DeepStar model as a control. So we basically fixed the number of model evaluations for NTv3 and for DeepStar. And for DeepStar, we did something like 500,000. We did a random mutagenesis of 500,000 sequences and used DeepStar to select the top candidates. So that's 500,000 model evaluations. For NT, we did something a little different. We used 50 steps per generation in the diffusion process, and so we generated 50 times less sequences, and then we used NT's own predictive head to select what it thought to be the top performing sequences of that 10,000. And these are the sequences we sent to be tested in the lab. Finally, I guess we have an ablation, which is in silico, on the impact of pre-training and post-training. So here we wanted to ask, uh, this is all great, but does it help to start from a pre-trained model? Or can you just get away with training this diffusion model directly on the enhancer data? And so we have a few experiments here that are interesting. What we find is that pre-training helps across the board. So if you look at KL, um, if we start with the pre-trained base, um, we see lower KL, which measures basically how closely the oracle predicted activity, I think in this case we use DeepStar, of generated sequences matches the held out data. Interestingly, we see another effect, which is that here we have the option of basically including the sequence context around the enhancer or not. We find that the sequence context around the enhancer only helps if you had started with the pre-trained base. And this kind of makes sense that if you have the pre-trained base and this kind of general knowledge, you're able to leverage the interactions between the enhancer and the surrounding context. But if you don't have that pre-trained base, then the surrounding context maybe obfuscates the task, and it actually hurts generative performance. So we thought this was a pretty interesting result. And yeah, so that's the end of the presentation, the main presentation. I have some kind of additional slides on ablations and things if people have specific questions. But yeah, thank you guys for having me. We've tried to open source everything as cleanly and easily as possible. So everything should be as efficient to use. And so yeah, we have kind of our repo here, and you can read the paper if you haven't, and check out the live demo, which allows you to we've hosted NT, so you can put sequences and get the functional and annotation predictions, as well as we have a variety of notebooks showing how to fine-tune NT, how to generate sequences with it, and some others as well. So yeah, thank you guys for your time. I think there's maybe been some questions in the chat.